So today we are going to go a little bit, really very briefly, on last few issues that I wanted to emphasize from last week. But mostly we will talk about, I'll mention ELIXIR, which is a very important and fairly new initiative that I don't know how many of you are familiar with, but I think it's worth knowing something about it. And then we'll go to Genome Browser. And throughout this discussion, I'll mention few major resources that I think it will be really worthwhile to know. So that will be the basic of the story. And let's start just by reminding, this is just a reminder on GenBank. We talked about it. And just to know that I mentioned GenBank as a resource that is a little bit messy, a little bit redundant, and so on. It's including these three different resources that are mentioned here, but I definitely want to mention that there is a point to go into the individual resource. And why is that? And I'll give you just one example from the EBI Ensembl database, because in each and every resource there is a little bit of idea what's behind. So for example, in the database of the EBI, they really care about weird organism, about metagenomics, about microbiome, and so on, while the other put less emphasis on. So you see a little bit of flavor for each of those. And at least I don't know that much about the Japanese, but at least the EBI Ensembl is the European Molecular Biology Lab, which is the equivalent of NCBI from many perspectives, provide really a beautiful database that at least I'll give you one example. Just to show you the obvious, that the growth in those database scale is enormous, really unbelievable. And this is just to show in, terabytes, in base pairs, in whatever, just for a reference. This is a human genome complete. As you know, it's not even complete, but let's say this is the human genome. So you see that this is a 10 genome, this is 100 genome. And so the scale is just enormous. Consider that most of the data comes from bacteria and viruses and so on. You see how many entities are in the game. So this is just to show you this exact subset of the Ensembl. It's called ENA, which is European Nucleotide Archive. And ENA, has its own release, and look at the scale. The scale is really, there are 261 million entities. Entities means organism units that are, and numbers that I cannot even read of nucleotide, and it's distributed daily. So if you just saw something that is happening, every day will be updated, which is kind of nice. The other something that I kind of like is that it's distributed daily, and they said we cannot really cope with the others, so don't rely on this being cross-referenced to any others. We move, and once in 3 months we are trying to do some coordination. So if you go there, be aware that it's not fully coordinated with the other unless there is a new release. So that's one point just to make it clear. Now the RefSec, we discussed quite a bit about it as a derivative database. And I think we already mentioned this. I just want to emphasize again, just so you'll be very familiar with the way you refer to the sequences. This is just for this reminder. So if you see XP, you know that it's what? Just, what is it? It's a protein, but is it something that I'm absolutely sure? I have seen it. It's X. What's the X means? Not exome, no. It's a model. We don't know. It's a model. So be aware that there is a difference between XM and NM. NM means nucleotide of mRNA that I've seen, and there is so it's not the same quality. And be aware that much of it's XM because it comes from machines. Okay, so that's another point not to miss. Now, the RefSec is linked to many good resources. We already discussed it, but I'm mentioning it because some of those resources are really the genome viewer and so on that we are going to discuss today. So it's really well connected. Okay. And as Nadav told you today, FTP is the right way to get it down. And this is, for example, the FTP of the RefSec, but you can do the FTP from many other things. So as I said, it's not I think I mentioned this before, but be aware that RefSec not always comes with the coordinates of the genome, right? It is a unit. I think I put emphasis on this last time, so be aware. But if you go to RefSec gene, you have the coordinates and everything is good in terms of genomics. So again, the names means a lot, okay? So that's also quite intuitive. Now, 2 minutes for ELIXIR. ELIXIR is an initiative that started about 6-7 years ago. Actually, I'm the head of ELIXIR of Israel for that matter, but ELIXIR is a pan-European unit, initiative for trying to make order and to make quality verification for everything in terms of infrastructure for biologists. So this is a very courageous project in which most of the effort is really to do standardization, coordination, formatting, reformatting, and again reformatting, and making it very easy for biologists, mainly also to make sure that things that have been developed and a lot of effort have been invested in will not die by themselves just because the postdoc moved, right? So there is this effort, European-based effort, that nowadays it's even going into very interesting direction of medical informatics that was not initially the purpose of ELIXIR. ELIXIR was really infrastructure-based. But the one thing that I really want to emphasize, it has maybe a quarter of its effort is on training. Namely, there are beautiful resources for training. For example, if you want to have an expertise in single cell classification, you just write in the bio tool of ELIXIR sequence-based classification, and you'll get all the training, 1-day training, 2-day training, workshop that have been done on these specific topics. So it's a beautiful resources that often we do not know. It's pretty fresh. Everything that I'm telling you is the last, let's say, 3-5 years, but it's very worth while to know about it. I just want to say, because it's built for the community, the largest community, doctors, agriculture, you know, everybody, it's built in a very simple and very intuitive way. Part of the effort of ELIXIR is to make it intuitive, so people would not have to dig in and try to understand what do you want from me, but instead just look at it and just I really want to mention it because otherwise all this effort, I mean, the power of this effort comes from the users. The users come and say, it's not working, or I didn't like it, or whatever. So it's an ongoing project that it's communication-based improving. So it has a chemical biology, it has a literature, enzyme, evolution, protein, proteome, gene, genome, but also services like computing, data resources, software tools, and core data of ELIXIR, what's considered by ELIXIR to be the best tools, you know, with the tag of Machon HaTkanim, with the tag saying, okay, out of these many tools, this is the one we support. And saying support means that they check that it's really working, it's really updated, and so on and so forth. So it's beautiful, really. And I must say that it's nowadays by far more successful than initially. The idea was really not to let things die just by themselves, but it became really powerful platform. Okay, this is it. then I want to go directly to the browser. Now, the browsers, again, they are, the idea of the browser, just the concept, is to be able to put all, and I say all, RefSec will be an example, but in a minute we'll see what is all, in a genomic context. So my analogy is like being in the desert, let's say, but with or without Waze, you know, GPS. So either you know where you are or you are lost. So this analogy is not totally unrealistic, because the coordinates of the genome are really the coordinates in which you can navigate back and forth. You have anchor sites and so on. So it's really navigation tool in a sense. And now the desert is not a desert, it's just a genome, which is as bad as desert, let's say. So as good as desert. So that's the idea. And there are, in general, we mostly talk about UCSC, but I want to take a few minutes to show you that there are basically 3 major browsers. All of them are good. They are not the same. Some are more intuitive than others. But again, like always, if you put the effort and dig a little bit, you'll find that some of those browsers works better for you than the others. And fortunately, again, last few years, they are coordinated. So you can jump from one to another in a fairly easy way. The one that we'll talk most about is the UCSC browser. But I want to take you into Ensembl browser. And Ensembl browser is the European browser, which is, I think, is a little bit harder to get in, but it's fantastic. Who is using the Ensembl browser? Yeah. Okay, so at least 2 of you. It's a very good one, and you have to put a little bit of effort to know it. And the other, when I say, it's either the organism-specific browser, which I won't talk at all about. So for example, when a genome center is working on the alligator genome, they build their own browser so that all the information relevant to the alligator will be there in a, but I don't suggest as a learning experience to go there, although some of them are really beautiful, really good. But there is now another version of the browser that is called the IVG, which is a very good one, and maybe I'll show you a snapshot of it. It is used by GTEx and other very fancy, broad-like resources, and I think it's worth to know them as well. So if we have time, I'll show you. Otherwise, I'll just mention it. So, but the 2 big ones are very important. So we start with something like that. You know, you worked in the homework and so on. There is this exome, intron. You have the chromosome, the location, and so on. It's not as fun as to look at it in a real visual a way in which you can navigate, and that's the idea. It's the navigation tools rather than, and visualization rather than information per se, because information, you have it kind of all in the sequence itself. And we play with both directions. So just a visualization, and we'll do some demonstration, but I want you just from Ensembl to have a idea what we'll see. So Ensembl do not show you everything unless you ask. That's the concept. So they are very limited of what they present to you. But if you ask more, no problem. So they rely on you rather than flood you with something that you don't want to look at, or it's confusing. And we'll see that UCSC, I think the default of UCSC is a bit overwhelming in my view. But this is really a matter of view. So they start by saying, this is an old version, but it doesn't matter. They start by saying the transcript, let's say if we are talking about gene, but it doesn't have to be gene, about transcript, and then immediately they see a gene summary, and the gene summary is something that you immediately see, and without having to dig deeply, you see if it's a big or small, you see if it's an exon or intron, you see the size of intron and so on. Immediately you get an overview including the length of this transcript. I'll give you an example, and that's basically where you start. This is the starting point. Now, if you are interested in how this gene looks in other organisms, no problem, we'll show you. Are you interested in what is the homologue of these proteins in the same organism, i.e. paralogs? No problem, we'll show you. So it's a very much waiting for your query to go on and deeper. And to tell you the truth, when you go on and deeper in terms of phenotype, variants, it's very rich and extremely good, extremely good. So it's not, don't be kind of saying, well, it's very dull. It's very deep. It's not dull at all, but it's not flooding you. That's the point. So I'll give you an example, but I want to mention, as I said on the Ensembl, you start by asking which organism I'm interested in. Here we tend to talk about either viruses or human, just because viruses are fast, quick, and human are complicated, and just as if nothing is between. But actually, those resources are phenomenal to people that are working on phylogenetics and on other organisms. So just despite the fact that now you can put, again, it's a very flexible machine because it's supposed to be open to everybody. So if you don't know, you just have a hint, what is this proteins? You don't know even the symbol, you just know some words about it, it's good enough. It will give you options and say, okay, I know that you don't know much. Let's see the list, and you pick from the list, which is very nice, because you can write RNA transfer. But you don't know what transfer, which RNA. It's good enough. It will give you immediately the subset of RNA transfer so that you can pick from that. That's another kind of easy way to get in. And maybe I'll skip this if I have time. This is, you know this, how many of you are familiar with this screen? No, not many of you? It's a beautiful screen when you just get into Ensembl, not necessarily Ensembl browser. You have a beautiful way, really beautiful way that breaks your biology into 5 big sections. So I plan to go with examples, but I'll just give you the header of those 5. The first one is really genome. This is what we are dealing with today. The second one is a beautiful comparative genomics. So all comparative genomics, how my sequence relate with visualization, with statistics, really nice. The third one is regulation, which is going back to ENCODE that I told you before, which is where is the enhancer, where is all those hidden, beautiful site. The very unique one that Ensembl actually takes the lead on is the gene annotation. What are those genes? And again, as you can see, we talked about it today, they give you several options of download. Do you want the FTP? Do you want this format already as a starting point? So feel easy with what this is the gene, and then the variant. And the variation, we'll have few talks about variation, so I won't explore it, but the variation already hooked to phenotype, to human phenotype or other phenotypes. So it's a very, very rich and very good portal to start with. Nevertheless, if we want to go directly to, this is an old version, maybe we'll go to the first demonstration. And this is just when we go to Ensembl, just to the portal you know nothing about, and you just write the name of a gene. In this one I wrote SIT1. SIT1 is synaptotagmin 1, sorry. Synaptotagmin 1 is a very famous, important gene that actually control everything that happens in your brain in terms of the link between neurotransmitter release and activation of the neuron. So this is the bigger switch, the most important switch in communication, in neuronal communication. And as you can imagine, there are a lot of diseases that are associated with it and so on. So it's a fairly amazing proteins by itself. So I kind of like it. And when you just in the web of the Ensembl, just write the name or any name, they give you 22 results. But immediately look at the left side. They say, are you interested in the gene, in the transcript, in the variant, in the phenotype? So they ask you, it's all communication. So let's say we are interested in the gene, because that's the topic of this class. So I'll go directly to the link, and maybe we'll go directly to, let's see. What do I have to do? Okay. If not, I'll just go directly to the browser. Okay. Okay, sorry. Okay, so let's see what we have here. So let's go again. As I did, we can do Ensembl. I'll go to Ensembl, and immediately I'm in this page. So let's go to this page and say, okay, I'm interested in this case in human, but as you can see, the list is, can you see it? Yeah, like this. Okay, so I'm interested in human. Oh, I'm interested in human. And immediately, this is the page that we discussed before. And here, you can just write this sit1 or any other. So immediately, it goes. And this is the page that I already showed you. Can you see it good enough? Is it? This is the magnifier. You can just sorry? Is it good enough? Okay, excellent. So let's say we are interested in the human gene. And now, once I ask the human gene, it brings me to a summary of the human gene, right? And in the summary, I say, this is the name, this is the location. And the location will bring me to the browser, of course. But immediately, it gives you some summary. You know, before, this is always a good idea to know where you're looking, just to get the orientation. So you have 12 transcript and such paralog and so on and so forth. And you have also few excellent resources. One of them is, of course, RefSeq, but it's not the only one. But now you can go directly to what I showed you before. This is the gene itself, isolated, not in the context of what's left and what's right. But as I said, we want the genome. We want to navigate. So I'll go directly to the chromosome. And now I'm already in the browser. And as you can see, I won't go too much into the browser. But as you can see, I'm in this synaptotagmin 1. It tells you immediately the direction of the protein, if it's going from here to there by the arrowhead. But immediately, you say, I want this area. And when you do this, you are in this area. And you say, you know what? I want actually to be in this area. And you see this, and you go directly to this area. So you can navigate just by marking without putting numbers, just by viewing and say, well, I'm interested really in the neighbor. Who is this guy that sits next to it and maybe shared some a common promoter or whatever, or enhancer or whatever. So it goes, and it takes some time, but it goes directly to what I selected as a focused area, right? So that's make it very easy. Again, as I said, I just want to say that you can just look at, as I mentioned, whatever you want on this, let's say, patch that I'm here now. And this is the size of the patch. As you can see, it's quite big. But you can say, show me this region, how is it in the mouse? So I'm asking the synteny, and the synteny just take whatever I was there and show me that this region, that it was in this chromosome, it split to this chromosome in the mouse. So this region that I started, this is what happened in the mouse. this region went to this region went to this, and I didn't even click a letter. I didn't ask anything. I just wanted some kind of an overview of what happened, how dynamic is this region. So you see, without talking much, how informative is playing with the pre-prepared list that they prepare to you. Amazing, right? So what I'm saying is that there are two ways to play with Genome Browser in general. The passive way, which is show me what you want to show me, and the active ways. And I'll demonstrate it in the UCSC, what is the level of communication with the browser that basically you can put your own data, you can visualize in the same way, you can store your data, you can share it to the public, and so on. So it's really a platform more than a browser in the active format of users. Okay, so today we won't talk much about the active part, but on the passive part. But I want to make a very clear statement about it. If you have an experiment and you want to show it in the paper, in the publication, in your PhD, in your master, or whatever, this is a very legitimate way to do. To add it, to add a track, we call it, and then you can see it. So I think I show you a little bit of the power of this method of Ensembl. And again, maybe I'll keep it for another time, but for people that do not know BioMart, it's worth to know because it's a very good downloadable entry. So you can download everything in define. So let's download all the genes from all mammals that are kinases. That's it. You click, you have a list of kinases from all organisms that you, so, okay, so this is a querying system, I think the best, that is really, really good and supported by Ensembl. This is their own query, so there are many good tools here. This is just to say. Now, after saying that, let's go directly to the browser, and let's go maybe from here to UCSC, and we'll do some demonstration. UCSC, and actually, when you do UCSC, it's a university, pretty big one, but Google doesn't know about this university. It's only about this browser. And they have a very nice, even here, a very nice blog showing how the university disappeared from, because you see the scale of users, and all the other are just on the bottom because of the scale. You don't see that anyone entered this university ever. And it's funny because if you just open the scale, you see that a lot of people are interested in this university, but it's very embarrassing. And the guy that is heading this Genome Browser has some sense of humor, so he always have a blog showing why the university is, disappeared and only UCSC remains. But actually, it's just a joke. Okay, so if we go there, let's. I think this page Nadav showed you. The one you can just a comment. You can go to the mirror. Namely, they duplicated. Sorry. Right now, we can do mirror of Europe, which is often faster, but or Asia. They did the 3 points so that for the European it will be faster, and usually we want to use it in the American because they sleep now, so it's better. So usually I'm playing with this, to make it faster. You think who is sleeping, Japanese or Asia, or and then you use. Okay. Okay, thanks. That's good. Okay, sorry. So we have this, as I said, the mirror is good when you feel that the machine doesn't work that fast, so just move to a mirror, one of the mirror, but that's a comment. You go from the genome, and we already know about the genomes, right? And today we'll do the human genome, but as you can do, as I said, there is a very long list of others. I want, and as I already mentioned, most of the people, even today, are using the HG19, but I think many of the new, let's say, databases and tools are using this. And just to show that, feel free to use both or either of those, because there is a very good cross-reference within the system. So don't just know where you are more than decide what you want. I mean, just to know. And there is a tool here that can change from one to another. Anyway, let's go there. And when you go there, as I said, you can do, as you saw, anything, whatever you want. So this time we'll call it synaptotagmin. And this is not its full name, of course, but I want to show you. And the one thing that while it's coming, I want to show you that okay, so this is the list of synaptotagmin. As you can see, there are many of them. You see the name, and what we specifically looked for, as you can see, is synaptotagmin 1, which, as I started in the beginning, in chromosome 12. But you see that immediately I see all the paralogs and everything. So the idea is the following. You can, on the browser, in the term of navigation, you can either use those that are very useful, so zoom in by the level you want, either by little, by 3 times, or 10 times. Because sometimes you start with a variation of 1 letter. So you are zooming into 1 letter. But immediately you can do double click on 100, 100, and you are already with 10,000 around you. So it's very easy to navigate without the need to go into decide, I need 50,000, I need 30,000. You can just use this. I think this is the most clickable way to navigate. So this is one point. And you can do zoom in, zoom out, back and forth as fast as you want. Now you can also do the move. And the move, you can do a small move. Namely, the frame will stay the same. You just move 50% of it, or a big move. And why do you want to do a move? Just give me an example. I'm interested in this protein or gene. For example, to understand gene around, what more? What could be in terms of what? Yeah, the regulation, where the promoter, where the enhancer. Could I see some kind of a very active transcriptional level and so on? So you have a lot of information by going out, zoom out and see the context, okay? So, but right now, as I said, I'm looking at synaptotagmin, and let's do 3, I just focus on, let's say, zoom out of 3 times, and okay, and I already, immediately it tells you what is the scale of the game, right? So I'm in a resolution now of 500,000. And what do I see? I see first the chromosome location. And as you can see, if I'll do 110, this one should grow. And immediately you see that it grows because it's live. And again, I want to zoom in into this protein, this gene, and immediately I can move it. As you can see, I can zoom in. Now, what's the code? First of all, the direction is written. How? By these arrows. I don't know if you see it good enough, but the arrows means that it goes on the complementary strand. Immediately you know it. Then, so this is the more 5' of the game, and this is the most 3'. Now, every let's say long line here, it's an exon. Every small line, and let's zoom in on this very tail of the protein. Let's do this just for, I can zoom in, you see? And now I'm already in 16,000 instead of 500,000, okay? And what I see here, this is an exon, this is an exon, this is an exon, but non-coding. You see? So they have a very simple and intuitive code. You don't have to read much to understand what you see, right? And this code is become very common in all browser. So why do we have different colors? We have different colors because the blue one is the refsec, the good one that we would like to follow if and only if we're interested in the major pre-decision transcript. Okay? So you can immediately, just by looking at the colors, decide where you want to see. And the point is the following, and stay with me because it's quite interesting. I'll do even a zoom in. The UCSC provide you a default of what they believe you are more interested in, which is, of course, how do they know? However, it's very, very easy to change the configuration of the page. And I'll show you an example. But think about the concept, and then you'll play with it, and it's very intuitive. So, for example, this is a gene code list. This is an refsec, and as they say, they have all those annotation list. And this is, they decided that you are very interested in expression. Now, it makes sense that you are interested in expression. But they provide you already a snapshot of expression that usually you don't know exactly what you're looking at. But if you don't know, you immediately click on the header and you'll know what you're looking at, which is very nice in textual way. And what you see here, I'll first just go without clicking just to show you what is the default when you didn't do anything. By the way, even if you did something and you come tomorrow, they will keep you the session, so whatever you decided to look as important will be the default in your computer, which is very nice. They are pretty clever in saying, okay, those guys are really interested in transposomes, so we won't show them transposomes. So somehow, by using, you get to know what you like, and you can keep it as your session as a default, which is, again, nice. There are many nice features here. Now, let's say you are more interested in the expression, so you just move it. So you can do even by just moving it, you can reorganize the page in terms of what's important, what's not important. Okay. But what you see here is a 53 tissues of GTEx, which is a very important and very useful, of 570 donors on 9,000 samples, and where the protein was expressed among these 53 tissues. And I think by staying on each, you can immediately see that there is this yellow patch, and this yellow patch is different part of the brain. Because when you stand on each, you see immediately where are you, and you can, although I said I won't click, I will click this, and immediately you see the resolution of this in a higher resolution of the same kind of snapshot that they give you. So in this case, you see that it's in amygdala, in the brain, and in cerebellum, and in the hippocampus, and so on. And you can see that it's not expressing outside of the brain, which makes sense. Okay, so this is and you see exactly how the expression is, including some kind of explanation, what is the display and how you can change the display. So if I go, how do I go back? Here, yeah, it's, okay. So what the next thing that they show you that is very interesting is what I told you about the encode, where and how the expression level in many different cell lines. So what you see here, this wave, this kind of wave, saying the following. If we have a 100 cases of, let's say, sorry, the scale is between 0 and 100, which of the cell line had this signature of chromatin that tells us that this area is actually active in terms of transcription. So just by looking at the wave, you can see that there is something here very dramatic in terms of enhancer, promoter, and so on. And I didn't do anything, you see. I don't even have to know something about the gene in order to focus on a specific region that I can, again, by clicking on this, I can say what I'm showing. I'm showing encode 7 cell line, and each cell line has a color. So now I can see what is actually the reason for this very high expression level. Very nice. The second, the other part that they believe are super important is the hypersensitivity, which means what areas are sensitive if I come with DNase. Now, DNase is an enzyme that breaks when the chromatin is open. So this is a very good proxy to activation, but it's not one-to-one. So immediately you have another track layer, which is part of the regulation, but this time is just shown as a color-coded between white, which is nothing, to black, which is very sensitive. And I don't think you have to be very clever to see that they connect very nicely, right? This track of the H3K27 acetylase and the DNase. So whenever you have strong DNase, you have also strong activation, which is beautiful, right? Look how much biology you have by one screen here. And then they have something that some people like, some people likes less, how this region is conserved in terms of, again, colors with 100 vertebrate. So I can show you more. And then, as I said, this repeats, sign, line, transposome, and so on and so forth. However, the most important part is this one, and below. So what you have here is, I'll go with you on this scale, but actually you can go also directly from here, from the configuration. So either I'll do it from the screen, but the configuration will give you the same in a bit easier way to navigate. So the first thing is the mapping and sequencing. So they give you about 20 ways to talk about the genome. What does it mean? Again, there is a very good help. So whenever I click, I don't know what it is, immediately I get this idea. So for example, do you want to show the centromere in 5 different layers? So each and every entity, they provide you 5 different presentation visualization, either full or hide, and then all kind of compression level, okay? So you have to know the order. So it's between, let's say, hide to dense, which is very dense, to screech, to pack, and to full. They had to improve the English in order to make these 5, but because they didn't want to do it 1 to 5, because no one will know what's direction, so they did it this way. However, it's very, very important. And why is that? The default of doing show me everything is a nightmare. Don't dare. Exactly as Nadav told you, don't just upload and know nothing what will be the impact of it. Be very careful about it. So this is a very easy way, because you can always start with the very packed way and see, okay, now it's not enough. And do it in a very easy way to know what level of resolution. So if you want to do full variation, you'll get the information on each and every nucleotide from 2,600 individual. It's a nightmare. You don't want it. So you have to be clever in what you ask, okay? So again, so this is the mapping. Very, very good. And actually, they added this interesting patch. Patch sequence means that people are working with some assembly, and then we have new patches to this assembly that were not in any previous reports. So they patched it so that you can have a more complete way. So you can use the patch or not use the patch. This is, you know, details, but it's very active and ongoing. You know, a month ago it wasn't there. So it's still ongoing. Now, the more important to our discussion for now, or for this class, is the gene and gene prediction. So we know that the RefSeq, we can look at it as a packed. This is their default, but you can do it as a full. And you can also try to see, on terms of genes, what others are saying. Namely, for example, the gene code. Let's say the full gene code. Let's say the full Swiss Protein Institute in Switzerland, which are very good themselves. And you can have the UniProt that we'll talk about later. We can do it as quick like this. And you can have all kind of gene and gene prediction. And you can do refresh. And once you do the refresh, they are not waiting for you to decide that you do submit. They said, OK. They are very quick in responding, saying, OK, that's what you want, have it. And immediately you see that in this specific case, it didn't change much. So the packed and not packed didn't add any type of information. However, if I just want to go to the, let's say, to the gene code or to the RefSeq, and I want to change it directly, I can, let's say, either hide it. I don't want to see it at all. Or I want to do this just as a dense. So what I see as a dense, I see only one that is a collapse of all the transcript into one. So as if I just collapse all the exon intron on one slot. So as you can see, maybe I'll show it again, because look at the RefSeq here. You see this exon, and you see this exon and 3' UTR. So let's move now just to do not dense, but pack. So when I see it pack, I see that this was a consensus between the two, and now they show it only as a one entity. But of course, it will be different if they were very different. So let me just go fast on what else we have here on the genome. So this is, as I said, other RefSeq means all the RefSeq of these proteins in other organisms. Okay? So this is something that you have to be very careful. So just do it as a dense. Don't dare to do more than that. And then you go to phenotype. This is, again, unbelievable importance, because now this region, it's not even the gene, it's the patch. Okay? It's the region that I'm looking at now. They tell me what phenotypes are associated in terms of disease, in terms of, as you can see, ClinVar, which is a combination of all the variants, the clinical pathological variant, in terms of cell lines that are information. And as you can see, most of it is hidden, because it's a very specific getting in, but I'll put maybe one of those. And then, just quickly, they move into the messenger RNA and EST. So what, as we've seen in terms of splicing, level of expression that I already showed you this. The GTEx I already showed you. Regulation I already showed you in terms of encode. Conservation I already showed you. And SNP I didn't show you because I didn't want. And repeats I can say, just don't show me. Let's say hide. And so on. Never mind where I do the refresh, they showed me exactly what I want to see. So immediately you see quite interesting. And the last thing I want to say, that while, or two last things. First, you can always click here and get all the information summarized in a textual way, in a very good way. Actually, you can go back to the browser. You know that the browser gives you 100 because you started with the browser. But you can really look at the NM ta-ta-tam, this is the RefSec. You have all the information, you have all the location, you have the predicted protein, you have description, and so on and so forth. This is one thing that I wanted to show you. That you can, the other things is that you can really go into table browser. Instead of visual browser, everything tabular. So the same information in a more, let's say, formatted and workable way. So if I just do this, I'll get all the information that I wanted on this specific region by click. So now I can say, send me as a file to this format, to this format, or just as an output whenever I want. And then I get everything that I talked about in a summary. And for that, you can also refresh your configuration. The last thing, that there is list of very good tools, extremely good tools. One of the good tools that I like to use is, as I mentioned the table already, but the BLAT. BLAT, you know BLAT? BLAT is a very useful tool. I guess everybody knows BLAST. So BLAT is a version of BLAT for a perfect match. What does it mean? It doesn't look for everything and do some heuristic, blah, blah, blah. It says, if you have a sequence which is more than 20, send it to me. And if there is a place in the genome, I'll show you. So it's a very, very good way to do fast, quick search within the genome. But you know that it's not, will open a lot of gaps. I mean, so immediately, if I'll just, this is very useful, the BLAT, and it's very easy. So for example, I can take the example here. Just, I'll throw it here. No big, I'm a Mac person, so okay, so I guess it's a copy like this, and you do like this, and let's do some mutation to make it harder for this guy. CC, and I do submit, and immediately it tells me, okay, I don't have a perfect match because I changed it, you see? But I'll tell you, you try to trick me, but actually we are here in this browser. So don't be so clever. We know what was there. So immediately, it's very easy, as you can see. And you are back to the same format in which you can config, you can do quite a lot. And often, if you, the only problem with this website is that it's very addictive. You start using it and you cannot stop. And often you find yourself in a different gene, different area, different connection. So you always can say, save me, take me back to the place I started. So it's also very helpful not to get lost in this desert, as I said. And I think I'll stop here. So I showed you, mainly I tried to show you the concept behind those browser, and really they are phenomenal. I think it's one of the strongest tools that we have in biology those days. In genomics, definitely.